Hariharan, you can start.
Good evening, everyone and all. I'm Hariharan from the Organizing of Medicine, and it gives me a great pleasure to introduce our distinguished speaker for today, Dr. Jagadish R, MBBS, MD, DNB, and currently the Assistant Professor in the Department of Biochemistry at AIMS Madurai. Dr. Jagadish has completed his MBBS from Government Teni Medical College, followed by MD Biochemistry from Christian Medical College, Vellore, and DNB Biochemistry from the National Board of Examinations, New Delhi. He has over 17 publications, including high impact work in iron metabolism, insulin resistance, and lipid biochemistry, and has presented national and international conferences. His academic excellence has been recognized with the President's Gold Medal in DNB, along with his multiple Best Research Paper and Presentation Awards. With this brief overview of an inspiring clinician and scientist, teacher, and laboratory quality leader, I warmly invite Dr. Jagadish R to share his insight with us in this evening. Thank you.
Hi, good evening. It's indeed happy to meet you all in this session. Before I start the topic proper, let me ask you two questions. Is my presentation visible?
Yes, sir, it is visible.
Yes, sir.
Is my voice audible?
Yes, sir.
Fine. Then we are good to go. Okay, fine. Thank you. First of all, I want to thank Dr. Raghunandan, Dr. Jamuna Satish for giving me this opportunity to present this topic in this forum. And this is a good initiative. Keep going, guys. Let's jump to the topic now. Okay, so before I start this topic, this is a really interesting topic that you guys came forward with. Thanks for that first. So this is what I'm going to speak today in this session. So let's start with this quote, which is said by Hippocrates. Let food be thy medicine and medicine be thy food. Unave marandu, marande unavu. Okay. So what we eat is actually determines what we become. So that's the core concept of today's topic. We are going to see this core concept in the context of cancer. So this is called, the topic deals with something called nutrigenomics and its role in cancer. So what is nutrigenomics? It is also called as nutritional genomics. It deals with two important things: how food affects our genes and how our genes will determine the response towards the food which we are taking. So it determines two important things. So for example, this is what it is. So what we eat, how it influences our genes, and how our genes influence the response towards the food which we take. So these are the two important concepts that is being dealt under the term called nutrigenomics, which is also called as nutritional genomics. So these are the principles of nutrigenomics. It basically determines the molecular relationship or interaction between the nutrient and gene and how it influences our epigenome, our gene transcription, our protein expression, and metabolism. So since it influences epigenome, nutrigenomics involves something called epigenetics. Since it influences the gene transcription, it also includes transcriptomics. Proteins, proteomics. Since it influences the metabolism, it is metabolomics. So nutrigenomics actually includes multiple omics under it. So nutrigenomics will, in overall, it can influence the overall health and susceptibility of a disease in an individual. So nutrigenomics is a broad term. So what is the link between the nutrition, which is the food which we take, and how it affects our genes? That link is established by naturally occurring biologically active compounds in the food. They are also called as phytochemicals, nutraceuticals, functional ingredients, and dietary protective factors. Okay, they are also called as dietary bioactives. So these compounds in our foods will influence the genes in our DNA. Okay, so these compounds are predominantly plant-derived compounds. Okay, there are few compounds from animal foods which can also act as dietary bioactive compounds, but predominantly they are plant-based. So these are the bioactive compounds or bioactives which is present in our diet. There are nearly 6 major class of bioactives in our diet. There are multiple classes. I'm just telling you the 6 major classes. The first class of bioactive compounds in our foods are called polyphenols, carotenoids, alkaloids and terpenoids, organosulfates, and polyunsaturated fatty acids. Polyphenols, you might have heard it quite often. Okay, it is the one which is present in tea, coffee, onions, turmeric, and soy. For example, these are the chemical names for those: quercetin, curcumin, epigallocatechin, epigenin, and resveratrol, caffeic acid. Carotenoids, you already know: vitamin A, beta carotene, which is present in green leafy vegetables and orange colored vegetables. Okay. Alkaloids and terpenoids, again caffeine, curcumin, and limonene, present in berries and citrus fruits. Organosulfates, very important group of bioactive compound, present in cruciferous vegetables like broccoli. Okay, they are also called, the compound name is called sulforaphane. Okay, then polyunsaturated fatty acids, which is mainly found in the fish foods, especially the fish which has a greasy and oily skin, which contains omega-3 polyunsaturated fatty acids and omega-6 polyunsaturated fatty acids. So these are the 6 major class of bioactive compounds in our food. So that is nutrigenomics. We know what is nutrigenomics, and we know what are dietary bioactives. Now we are going to see cancer. Okay, so cancer, when you say it's the leading cause of death worldwide, accounting for nearly 10 million deaths in 2020, as per the latest WHO fact. Okay, so there are 4 major factors which is primarily associated with cancer. What are those? Genetic mutations involving the genes, especially oncogenes and tumor suppressor genes. Exposure to carcinogens, that is, carcinogens are the agents which predisposes to cancer, okay, which can be in your diet in the form of preservatives, smoked, salted, spicy food, okay, which can be in the air. Okay, poor quality air contains a lot of carcinogens, pollutants. Okay, then it can be some drugs. Okay, X-rays. Okay, so these are the carcinogens. Okay, prolonged inflammation in a chronic disease. For example, somebody is having prolonged hepatitis. Okay, they are more prone to develop hepatocellular carcinoma and oxidative stress. So these are the 4 major factors involved in the or primarily associated with cancer. Okay, fine. So when you say cancer, there are nearly 10 hallmark features for a cancerous cell. Okay, so what are the 10 hallmark features? They have sustained or uncontrolled proliferative capacity. They keep on multiplying in your body. The cancerous cells can keep on multiplying, and they can evade growth suppressing signals, and they can evade apoptosis. That is, they can resist cell death, and they can also avoid immune mediated destruction. That is, when a cell is produced in your body, okay, your immune system will attack it. Okay, that's a normal response you expect, but cancerous cell will escape that. They escape that immune mediated destruction so that they can survive longer, so they can multiply further. Then replicative immortality. Okay, they'll keep on dividing. Their DNA gets keep on replicating. Their DNA will not stop replicating. That is, cancer cells have some mechanisms to do that. Then tumor-associated inflammation, very important one. Okay, whenever there's a tumor, in the tumor microenvironment, if you see, there will be inflammation. That inflammation is necessary for tumor progression. The inflammation is necessary for tumor metastasis. Okay, then angiogenesis, formation of new blood vessels, which is a hallmark feature of cancer. Then modulating cell metabolism. The cancerous cell metabolism is unique and distinct from the normal cell metabolism. So these are all the changes that happens in a cancerous cell, and these are the hallmark features of cancer. Okay, now the nutrigenomics, that is a food which we eat. Okay, I said nutrigenomics is nothing but how the food causes effects on a gene. Our foods, especially those 6 groups of compounds I showed you, they all have some effects on these 10 hallmark features. Okay, that is, the food which you eat can have anti-cancerous properties by modulating these 10 hallmark features. That's what we are going to see now. Okay, fine. For example, first, before going into that, let's see about few things. What are oncogenes? Oncogenes are produced in our body, are present in our body as proto-oncogenes. Proto-oncogenes are normal. Okay, proto-oncogene is a segment of the DNA which codes for a protein which is responsible for normal cell division. Okay, proto-oncogenes, normal cell division. When this proto-oncogene become an oncogene by a mutation, that is where the problem will come. What happens is, this proto-oncogene will become oncogene so that the cell will divide in an uncontrollable manner, leading to cancer. So proto-oncogene conversion to oncogene is a problem. Okay. For example, these are the proteins or these are the genes which can become oncogene if they are mutated. For example, cyclin proteins. They're responsible for cell cycle. Cyclin protein, cyclin D1, E1, A, and B. They are responsible for moving the cell from one phase of the cell cycle to the next phase of the cell cycle. When they get mutated, what will happen? The cell cycle will happen faster. Cells will divide faster. It is nothing but hallmark feature for cancer. Uncontrolled proliferation. Epidermal growth factor receptor. Correct. When the receptor gets mutated, it will keep on sending the signaling. So the cells will proliferate in an uncontrollable manner. Okay. HER2 receptor, which is the one which is involved in breast cancer, and BCR-ABL fusion, which is present in CML. So these are the examples of oncogene, how a normal gene becomes a non-oncogene with the help of a mutation. Then comes the second set of genes called as tumor suppressor gene, as the name signifies. Okay, these are the genes involved in suppressing the tumor. That is, they either repress or inhibit the cell cycle, or they will promote apoptosis. What is apoptosis? Apoptosis you already know. Apoptosis is a programmed cell death. Once the cells complete their lifespan, okay, they will undergo apoptosis. Okay, fine. So this is a normal tumor suppressor gene. When it is present, it will produce growth inhibiting protein. So cell cycle or cell division will be under control when it is normal. When your tumor suppressor genes are normal, your cell cycle or cell division will be under control. So how do they do it? They inhibit certain signaling pathways. They inhibit pro-growth signals, such as they inhibit some angiogenetic factors. They'll inhibit metastasis and they will promote DNA repair. Okay. So what happens here is when this tumor suppressor genes undergo a mutation, what will happen? Cell cycle is not under control. It keeps on multiplying. What does it lead to? That will lead to cancer. Okay. So mutation of tumor suppressor gene will cause uncontrolled proliferation of the cells, which leads to cancer. I'll give you a few examples. P53, PTEN, APC, and BRCA1 and BRCA2. These are the tumor suppressor genes. When these genes gets mutated, cell cycle will not be under control. Cell cycle will be aberrant. So proliferation of the cells will be in an uncontrollable manner, which will lead to cancer. Okay. In this, we are going to see about one important tumor suppressor gene called P53, which is also called as the guardian of the genome or molecular policeman. Okay. It is the heart of the cell's tumor suppressive system. It is located on chromosome 17 in the arm P at the 13th loci. Okay. It encodes a 53 kilodalton protein. That's why it got its name P53. Okay. So when this gene gets translated into a protein, if you weigh the protein, that molecular weight of the protein will be around 53 kilodaltons. That's why it got P53. Okay. So why P53 is considered it's a guardian of the genome? Because these are the things which can activate P53. For example, whenever there is a DNA damage, which can be due to any reason. Okay. When oncogene gets activated, or when the cell is under a stress, for example, in terms of hypoxia or low nucleotides. Okay. Cell is not getting its enough DNA. These are the cell stressors. Okay. This all will activate P53. When P53 gets activated, what will happen? It will cause cell cycle arrest. It will not allow the cells to multiply further. Or it will push the cells to senescence. What is senescence? Senescence is again cell cannot multiply. Okay. Or when there is a DNA damage, P53 can activate mechanisms to repair the damage. That is called DNA repair. If the damage cannot be repaired, what will happen? It will push the cell to apoptosis. That's what is happening in the third. Then it can also inhibit angiogenesis. So that's why P53 is called as a molecular policeman or guardian of the genome. Okay, fine. So these are the activators and these are the downstream effects of activated P53. Basically, it will cause cell cycle arrest and will not allow the cells to multiply further. Okay, now we know nutrigenomics, now we know cancer. Okay, so what is the link in between the two? There are numerous studies which have explored the relationship of nutrigenomics and cancer. It has been shown that the dietary bioactives, which I mentioned earlier, those 6 major classes of molecules, they have some anti-cancer properties. And they employ various mechanisms to achieve this. Okay, fine. For example, the dietary bioactives, for example, the polyphenols, alkaloids, terpenoids, okay, and the sulforaphanes present in broccoli, Okay, they all can influence your epigenome. Okay, they can cause epigenetic changes. Okay, they can cause alteration in the gene transcription. They can act as an anti-inflammatory molecule. They can act as an antioxidant molecule. Okay, so when I say epigenetic changes, that means the epigenetic changes are mediated by modification of histones and methylation of DNA. Gene transcription, it basically causes regulation of transcription of tumor suppressor genes. Okay, we are going to see one by one. For example, dietary bioactive molecules modulate epigenetic changes, especially polyphenols, which is found in your tea, coffee, and other vegetables. Okay, they are reported to influence epigenetic changes such as they can cause DNA methylation or DNA demethylation, histone acetylation or deacetylation. They can also regulate genes responsible for expressing a special type of RNA called miRNA. Okay, so these epigenetic changes, what they will do is they will alter the chromatin assembly. Okay, that is, they will cause chromatin remodeling. Okay, chromatin remodeling is a important step to that will determine whether gene can be expressed or not. Okay, fine. For example, this is what I'm trying to tell you. Okay, you see the figure on the right side. Okay, what you see here is a chromosome. Chromosome is nothing but the DNA assembly. So if you go deeper into the chromosome, this is how it will look like. These are the strands. These are called as chromatin fibers. Sorry. Sorry. Okay, fine. So these chromatin fibers will will be condensed to form chromosome. So if you see carefully, this chromatin or the DNA will wind around a protein called histones. Histones are basic proteins or positively charged proteins. Okay. So the DNA will wind around these histone proteins, and this histone will condense together to form chromatin. Chromatin will condense together to form chromosome. Okay, this is how chromosome is assembled. Okay, so what happens here is, since histones are positively charged, DNA is negatively charged, so they coil around each other. Okay, so what happens is, if the DNA is tightly packed, that is called heterochromatin, the transcription will not happen. Okay, if the DNA, this chromatin assembly becomes relaxed, that is called euchromatin. Okay, where the gene transcription will be activated. Okay, so how this happens? When it will exist as heterochromatin, when it will exist as euchromatin, or what can cause the shift between these two? The shift can be caused by two things. Okay, chemical modification of histone proteins, which I told you here, those basic proteins. When you chemically modify this histone protein, this assembly can get disturbed. What is that chemical modification I'm talking? You can either acetylate the histone or deacetylate the histone. What is acetylation? You are adding positive charge. Sorry, you are adding negative charge to the histone so that the positivity will come down. So what will happen to the assembly? It will get relaxed. The reverse will happen in deacetylation. For example, if you go to the next slide, you will understand it better. Okay. For example, let's say these are the histone proteins. What you see is those blue color circles or balls, spherical things are histone proteins. And what you see on this, this black colored strings are the DNA. The DNA winds around these histone proteins. DNA is negatively charged. Histones are positively charged, so wind around each other. Okay, fine. This is tightly packed. This is called inactive chromatin or called closed chromatin. Okay, fine. Now, histone is getting acetylated with the help of an enzyme called histone acetyltransferase. What will happen? It will add negative charges to the protein. So the chromatin will become loose. Chromatin will become relaxed. Okay, this is called active chromatin or euchromatin. It's called open chromatin. Okay, so acetylation of histones will result in the formation of an active chromatin assembly. So the DNA is relaxed. Now the transcription factors or the proteins which are responsible for transcribing a gene comes and binds to this DNA. So gene transcription will be activated. You understand? This is what happens exactly in acetylation. Okay. And those food items which I told you, curcumin, which is found in turmeric, ellagic acid, epigallocatechin, which is found in the tea. Okay. They are of all can cause acetylation of histones. So they can activate gene transcription. Okay. They can activate the transcription of tumor suppressor genes. You understand? So that's how the food items can cause epigenetic changes by chemically modifying the histone proteins, thereby converting the heterochromatin to form euchromatin, inactive chromatin to form active chromatin, thereby switching on the transcription of the genes which are involved, especially tumor suppressor genes. Okay, this is what happens when histone gets acetylated. Okay, fine. Now, what happens if the reverse happens? If histone is getting deacetylated, what will happen? Can somebody tell me what will happen if histone gets deacetylated? Now you're removing the histone molecule, acetyl groups from the histone molecules. What will happen to the structure of chromatin? Will it become activated or will it become inactive? Exactly the reverse will happen. Okay. The active chromatin will become inactive chromatin. Okay. That is done by an enzyme called histone deacetylase. So when the transcription gets inhibited. Okay, fine. So that's what exactly happens. So this is histone deacetylation. Acetylation activates it. Deacetylation inactivates it. So this histone deacetylase will be inhibited by those molecules, so that the tumor suppressor gene expression will be, transcription inhibition will be inhibited. You understand? So it activates acetylation of histones, thereby activating the transcription of the genes involved, especially the tumor suppressor genes. So that's how these food molecules can cause epigenetic changes in the genes involved in cancer metabolism, cancer progression. Okay, one. Next. The second epigenetic change can be modifying the DNA itself. Okay, what we have seen so far is modifying the histone protein, the protein around which the DNA coils around it. There are some modifications happens in the DNA itself. For example, methylation. Okay, methylation happens only in the certain specific areas in the DNA. They are called as CPG islands. Cytosine phosphorylated guanine islands. Okay, and those regions can get methylated. Okay, and those islands are mostly found within the promoter region of the DNA. What is promoter region? Promoter is nothing but the sequence of DNA which binds the transcription factor. It's responsible for initiating the transcription. So what is transcription? Transcription is nothing but converting the DNA to form RNA. Okay. For the first step is to initiate the transcription. How to initiate it? The transcription factor should come and bind to the promoter region. And those promoter regions contains the CPG residues or CPG islands. Okay. So what happens here is when the CPG islands are unmethylated, the gene expression will be active or the gene transcription will be active. Okay. When it undergoes methylation like this, when especially the cytosine molecules undergoes methylation to form 5-methyl cytosine, then the gene expression will be suppressed. Okay. So methylation will switch off a gene. Demethylation will switch on the gene. Okay. This methylation and demethylation can also be brought about by those dietary bioactives, especially polyphenols. Polyphenols can cause DNA demethylation. So when the DNA gets demethylated, what will happen? The gene transcription will be activated. You understand? So that's about the second epigenetic changes that can be brought about by those food compounds, dietary bioactives. Okay. They can cause DNA demethylation. Okay. That is done by an enzyme called DNA. Methylation is done by an enzyme called DNA methyltransferase. Okay. Demethylation is called DNA demethylase. That's it. So this polyphenols will activate the DNA demethylase. Okay, fine. When it comes to methylation, there's one important micronutrient which you should remember. That's called folate. Okay, methylene tetrahydrofolate reductase. Okay, that is encoded by a gene called MTHFR gene. Okay, is an enzyme involved in methylation and can influence DNA synthesis and DNA repair. Okay, this is nothing but folate, folic acid, vitamin B9. Okay, so that's why there are many studies that have shown folate has has a significant role in carcinogenesis, especially in colorectal cancer. Okay, they have a protective effect against colorectal cancer. So where is folate present? Folate is present in green leafy vegetables. Okay, so if you take a lot of green leafy vegetables, that is actually protecting you against colorectal cancer. Okay, this is one of the example. Okay, fine. So this is one more slide to explain that bit further. Okay, for example, DNA methylation in cancer. CpG island, when it gets hypomethylated. Okay, when it gets hypomethylated, that means it undergoes hypomethylated means the gene transcription will be active. When it undergoes methylation, what will happen? That gene will be switched off. If it is the gene of a tumor suppressor gene, what will happen? Tumor suppressor gene will be switched off. What will happen? Then there will be cancer, because tumor suppressor gene should be normal. When it is normal, the cell cycle or cell division will be under control. When the tumor suppressor gene is not active, then the cell cycle will be aberrant, leading to cancer. One thing. Okay. Then hypermethylated perisyndromic region, that is in the heterochromatin form. When they get hypomethylated, then they will come genomic insufficient cancer. This is how methylation can cause cancer, especially the DNA methylation. Okay, fine. Then the third epigenetic change is microRNAs. Okay, these microRNAs are small non-coding RNA molecules. Okay, they bind to mRNA. Okay, what is mRNA? mRNA is the one which gets translated to protein. Okay, so these microRNAs will go and bind to mRNAs. Okay, when this binding happens, microRNA, mRNA, when this binding happens, that mRNA will be degraded. So what happens when mRNA is degraded? When mRNA is degraded, there is no protein. Again, that is also one of the epigenetic change that can be mediated through microRNAs. Almost 60% of the genes are in us are regulated by miRNAs. Okay, this is what exactly I told you. So there's a microRNA gene which forms microRNA, and this microRNA can go and bind to mRNA and it causes degradation. Okay, and this microRNA gene will be activated by polyphenols. How that will be activated? Again, the same thing. Acetylation, deacetylation, methylation, demethylation. That's it. So through those epigenetic changes, these polyphenols or other dietary bioactives can also influence the expression of miRNA. Okay, fine. So that's about epigenetic changes. So to conclude this first part, okay, polyphenols can bring about epigenetic changes mainly through histone methylation and modification of DNA methylation, and they regulate the expression of tumor suppressor genes, sometimes oncogenes, KRAS, which is a very important oncogene. Okay, then it can also regulate the genes involved in autophagy. What is autophagy? Autophagy is nothing but it's a programmed cell death. It's a kind of apoptosis. The cell has some machinery to remove the damaged organelles through a process called autophagy. This is also called as a type 2 programmed cell death. Okay, and it can also bring about the changes in the miRNA expression. Okay, thereby, by doing these epigenetic changes, these polyphenols and other compounds can exhibit anti-cancer properties. Okay, so that's about the epigenetic changes. Okay, fine. Hope the speed is okay. Fine. Can I go with the same? Can I go with the same pace?
Yes, sir.
Okay. Fine. If you have any questions, please put it in the chat box.
Okay, sir.
If there are no more questions, we can just proceed. Okay. Okay. Because that's the core concept of this presentation. Then we'll go on to some few more slides, which are a little bit lighter in concepts. Okay. So if you understood this better, then we can go to the next. Okay. Is there any questions? Please put it in the chat box, or we'll go to the next slide.
Well, as of now, there is no questions. Yes, if we have some questions, will be told in the end of the session.
Can you please speak a little bit louder?
Sir,
I can't.
as of
Does
now, there
anyone
is
have
no
any
questions,
questions?
sir.
Okay.
We'll be telling it in the end of the session if there is any questions.
Yes, please.
Okay.
Okay.
Okay, sir.
That sounds good. Let's
Okay,
go
sir.
to the slide then. Yeah. Thank you. Okay, fine. Now, one more thing which is very important is, as I told you, there are four features that are primarily associated with cancer. One is mutations, primarily involving oncogenes and tumor suppressor genes. Okay. Fine. Next, I told you there are carcinogens, exposure to carcinogen. Third, oxidative stress. Fourth, inflammation. Okay. So the dietary bioactive molecules have the capacity to detoxify or scavenge the free radicals in your body. Okay. That is, they can act as antioxidant. Okay. Polyphenols, carotenoids, flavonoids possess a very important property called antioxidant activity. So what is antioxidant? These are the agents which can remove or scavenge the free radicals in your body, especially reactive free radicals, especially the free oxygen species, which are very detrimental to your cell. How it is detrimental? For example, when the cell has a lot of reactive oxygen species, it can cause DNA damage. Is it good or bad? It's bad. Okay, damaged DNA is bad. It can lead to a cancer. Or it can cause genetic mutation. It can cause a base change, or it can cause a deletion. Okay. Then the reactive oxygen species can activate this enzyme called DNA methyl transferase. What this enzyme will do? This enzyme will go and methylate the DNA. Where it will methylate? It will methylate at the CpG islands. When the DNA gets methylated, what happens to transcription? Transcription will be inhibited. Okay. When the transcription, what happens if it is a tumor suppressor gene? Again, that's a problem. So DNA methylation causes inactivation of tumor suppressor gene. That again leads to cancer. Okay. Then the ROS can also cause post-translational modification of a protein. For example, it can cause ubiquitination of a protein. Okay, what does it will do? Ubiquitin is a molecule when it gets tagged to a protein or it gets tagged to a peptide. This ubiquitinated protein can undergo degradation in the proteasome. So protein will be lost. So without protein, cells cannot function. What if it is a tumor suppressor protein? That can again lead to tumorigenesis. So reactive oxygen species or free radicals are very bad to your cell because it can damage the DNA, it can cause mutation, it can cause problem in the DNA transcription, it can also cause post-translational modification, can cause proteasomal degradation of protein. All these can culminate in converting a normal cell into a cancerous cell, which is called as tumorigenesis. So the polyphenols have excellent antioxidant property. So what they will do, they will scavenge this free radical. So they'll reduce the levels of reactive oxygen species in your body so that these untoward or detrimental effects can be avoided in your cell. That's why they say polyphenols are good because they act as an antioxidant. You understood? So this is one more important property which makes these dietary bioactive compounds to have anti-cancerous property. Okay, fine. Second, third, if you remember carefully, the one important hallmark feature of cancer I told you is somehow these cancerous cells have mechanisms to escape the immune-mediated destruction, which is mounted by the host. Okay. So they escape this host-mounted immune response and they'll continue to proliferate in your body. That is the cancerous cell. Okay. This dietary bioactive compounds can have properties to boost the immune-mediated destruction of cancer cells. How it does? They can promote proliferation or promote differentiation of CD8 plus T lymphocytes. Those are nothing but cytotoxic lymphocytes. And they can promote differentiation of natural killer cells so that they can remove or they can cause destruction of immune suppressing macrophages and T regulatory cells, also called as Tregs. Okay, so these dietary bioactive compounds can promote differentiation of CD8 plus T lymphocytes and natural killer cells. Okay, they can induce production of anti-cancer cytokines, for example, tumor necrosis factor alpha, interferon gamma. Then they can inhibit one important cell signaling molecule responsible for immune-mediated destruction. It's called transforming growth factor beta signaling. Okay, so when this pathway gets inhibited, the cells will become sensitized to immune-mediated destruction. This all can be brought about by those dietary bioactive compound. Okay, so they can boost your immune system to eliminate or to cause destruction of those cancerous cells. Fine. Next. And the fourth important feature I told you is chronic inflammation can lead to carcinogenesis. For example, the best example I told you is hepatitis. Okay, if somebody has hepatitis for over a long period of time, they can develop hepatocellular carcinoma. If somebody has inflammatory bowel disease, they can develop colorectal carcinoma. Because prolonged inflammation can cause carcinogenesis. So there are certain dietary compounds which are anti-inflammatory in nature. For example, best example is polyunsaturated fatty acids. That is those fatty acids present in that is omega-3 and omega-6 present in those fish, which has this oily skin or greasy skin. Okay, this oily fish has a lot of omega-3 fatty acids, which are anti-inflammatory in nature. Okay, how they are anti-inflammatory? Because they are known to suppress something called nuclear factor kappa B signaling. Okay, this is the signal. This is a pathway responsible for promoting inflammation in your body. That is, they will promote production of inflammatory molecules or inflammatory mediators like tumor necrosis factor alpha, C-reactive protein, interleukin 6. The one more pathway which I have not mentioned here is JAK-STAT pathway. I think it's coming in the next slide. Okay, JAK-STAT pathway. There are two pathways which are involved in promoting inflammation. One is called NF-kappa B, nuclear factor kappa B, and Janus kinase STAT pathway, JAK-STAT pathway. So polyphenols, POFAs, and flavonoids can inhibit this signaling pathways, thereby bringing down the levels of those inflammatory mediators, thereby relieving or protecting the cells from progressing to a tumor formation. Okay, that's about this dietary factors which can act as an anti-inflammatory in nature. Right. Next, there are certain compounds which are responsible for which can regulate cell cycle growth as well as angiogenesis. What is angiogenesis? Formation of new blood vessels. Okay, so there are certain compounds in the food which can prevent metastasis and which can prevent angiogenesis. Okay, they basically does this by regulating certain pathways in your cell responsible for cell growth and differentiation. One of the important pathways is PI3K AKT pathway. I'm not going into detail of these pathways because it will be beyond the scope of this session. Okay, at least know the name of this pathway. PI3 AKT pathway, then Wnt beta catenin pathway. These two pathways are involved in cell growth, cell differentiation. Somehow, some compounds in your food, especially the curcumin, okay, are known to regulate these pathways. And epigallocatechin, which is found in the tea, green tea, they can regulate this pathway and keep the cell cycle under control. Okay, and JAK-STAT pathway, which I told you earlier, one of the important inflammatory signaling pathway. Okay, that can also be controlled by certain bioactive molecules. Then VEGF and HIF1 alpha. VEGF stands for vascular endothelial growth factor. HIF1 alpha stands for hypoxia inducible factor alpha. Both these factors are very important for angiogenesis. Okay, certain bioactive compounds, especially the epigallocatechin, can regulate this VEGF HIF1 alpha signaling pathway so that they can regulate the angiogenesis. So they can inhibit the angiogenesis happening in the cancerous cell. Okay, so that's how these dietary active compounds are acting or manifesting their anti-metastasis and anti-angiogenic property. Okay, both of which are hallmark features of cancer. Okay, right. Next. Okay, there are certain compounds which are shown to regulate the apoptosis of the cell. Okay, what I'm talking is about the cancerous cell, not about the normal cell. Okay, what these dietary compounds will do to the cancerous cell. Okay, so they will promote apoptosis of the cancerous cell by increasing the expression of pro-apoptotic proteins, especially BAX and caspase 3. Okay, if you see the apoptosis signaling molecule, okay, this BAX, you will see BAX, you will see BCL2, okay, and you will see executioner caspases, so on. So they will promote transcription of these proteins, pro-apoptotic proteins, okay, so that the cancer cells will go for apoptosis. At the same time, they will downregulate BCL2, which is an anti-apoptotic protein. Okay, fine. and they can also cause cell cycle arrest. For example, alkaloids, for example, vincristin, vinblastin, can cause distortion or disruption of microtubule assembly. If you remember, during mitosis, in the cell division, during mitosis, there will be mitotic spindles which will separate the chromosomes on either on towards the end of towards the cells, towards the cell. That microtubule assembly can be disrupted by certain molecules, especially vincrustin or vinblastin. Okay, so they can also cause cell cycle arrest. So that's how. So when you do that, what will happen? Cells will not divide further, will not divide properly. What will happen to that cell? Cells will undergo apoptosis. Okay, so that's how those dietary bioactive compounds can have or promote apoptosis of the cancerous cell. Okay, fine. Next. So I think I almost covered almost all the mechanisms involved. Okay, so to just recap. Okay, so these bioactive compounds comes under 6 major classes. Okay, and these bioactive compounds can or will influence epigenetic changes. First point, by causing DNA methylation, demethylation, histone acetylation, deacetylation, and they can also cause or regulate the expression of miRNAs. miRNAs can degrade mRNA, and thereby they can cause epigenetic modification of the gene involved, which can be a tumor suppressor gene or it can be an oncogene. Okay, fine. Next, these bioactive compounds has properties to boost immune-mediated destruction of pancreatic cells, and these compounds can have anti-inflammatory property, and these compounds can promote apoptosis. Okay, and they can also inhibit metastasis, and they can also inhibit angiogenesis. Okay, that's how these compounds have anti-cancerous property. So what are the implications? The overall implication of this is based upon this genomic interactions. Okay, based upon this, we can or there are attempts to devise a personalized nutritional plan to cancer patients. Okay, thereby their tumor biology can be modulated, their DNA repair can be supported to improve the response towards the treatment, to improve the response towards chemotherapeutic drugs, to reduce the toxicity of chemotherapeutic drugs, or to manage cancer-related metabolic changes. Okay, have you heard of Warburg effect, which is a metabolic adaptation shown by the cancerous cell? Okay, that Warburg effect can itself be disturbed by certain dietary molecules, so that the metabolic adaptation is lost, so cancer cells cannot survive. Okay, there are a few papers that have shown that. Okay, but here this is for personalized. It varies from patient to patient. Okay, is it easy to do? It is not easy to do. You cannot devise a personalized nutrition plan to a cancer patient because these are the challenges. Okay, we have strong evidence on what is the dietary pattern we follow. That means whether you are taking high carb, low carb, high protein, low protein, it's a mix. Dietary pattern is a mix of everything. There are studies to show, or there are studies to which has shown this is the dietary pattern of North India, this is the dietary pattern of South India, this is the dietary pattern of Western India, this is Central India, like that. This is the dietary pattern of US, this is the dietary pattern of Europe. Okay, but however, we do not have much evidence on individual nutrient intake. So without that, we cannot devise a personalized diet plan. Okay. Plus, this is an upcoming discipline. You have to do a genomics work in a patient. You have to identify the genes, what they have. Okay. What are the polymorphisms they carry? Okay. How with that you can correlate how they will respond to this diet. Okay. Whether they will utilize this food or whether they won't utilize this food. Okay. So this is an upcoming study. It is costly and more technical expertise is needed. Okay, so these are the challenges that is in this field. Okay, so with that, I have concluded. Okay, so these are the challenges and these are my references. Okay, so basically, I prepared this talk by referring to multiple articles. So please, if you have any questions, please ask them in the chat box.
All the participants are asked to drop your doubts in the chat box, please. room. Yes, Tarun, you can speak now.
Yes.
Tarun,
Good evening, sir.
Yes, yes.
Yes, sir. So
Good evening.
recently I have studied. I saw a research paper which
Yes.
is from United States Food and Drug Administration, which is like India's CDSEO. So they
Okay.
have just published that they have fast-tracked inhalable gene therapy, which lets lung cancer patients breathe in a tumor shrinking treatment. So when I saw this full today's seminar, like today's the teaching.
Mm.
So what my doubt is, so this gene therapy, whether the dietary bioactives could theoretically enhance this immunotherapy response. That was my first doubt. So can we enhance this immunotherapy treatment by using these bioactive methods, dietary bioactive methods?
First, I want to tell you that I have not read that paper. Okay, but
Okay,
the question is,
okay.
well, your question is, yes, we can. There are few papers that have shown that putting cancerous patients on a particular type of dietary regimen will has shown to enhance the immune mediated destruction of those cancerous cells. Yes, there are a few papers which have shown that. But I don't know whether in the context of gene therapy it will apply or not. I don't know. I have not read that paper. But there are few papers which have shown that putting cancerous patient on a diet regimen can influence their treatment outcome. But there is a recent meta-analysis on this, which I read it, which I can share the link maybe. That has shown, yes, there are benefits. They are responding well to the chemotherapeutic drugs with the personalized diet plan. But we need large scale studies to confirm that thing. It was done in, I think, Denmark. I can send you that link of paper link to you. Maybe I'll put it in the WhatsApp group.
Sure, sir.
Hmm.
And another doubt is whether when we combine this metabolic modulation
Hmm.
with these kind of gene therapies, so that in future, can we
Hmm.
expect the improved outcomes for the cancer patients? So when we are seeing recent, for example, when we are taking a Tamil Nadu itself,
Hmm.
past year it has been registered 1 lakh cancer cases. So
Hmm.
in future, with this modulated metabolic modulations and
Hmm.
our gene therapies now, which is developing more,
Hmm.
can we expect more improved outcomes for the
Yes,
patients?
yes. See, cancerous cells, they will have some metabolic adaptations. For example, that's what I told you about with that Warburg effect. Now, so that is one of the classical metabolic adaptation expressed by the cancer cells. Even though you have a lot of other nutrients, they will rely mostly on glucose. So they will take up a lot of glucose, irrespective of whether oxygen is there or not. They will convert all the glucose to lactate. That is persistent anaerobic glycolysis that happens in the glycolysis. That happens in the cancerous cell. So if you disrupt that metabolic adaptation by using some molecules, if you disturb that metabolic adaptation, they cannot survive. They will undergo for apoptosis. So that's why if you see during COVID, it's not in the context of cancer. During COVID, they were trying this deoxyglucose. That's mainly to starve the cells. So if you disturb the metabolic adaptation, if you disturb that Warburg effect, the cancer cells will starve. So cancer cells will not multiply further. Yes, immune modulation, the metabolic adaptation, if you disturb it, it is actually one of the areas where a lot of work is going on. A lot of papers are there where they have shown that when you disturb this metabolic adaptation expressed by the cancer cells, they will undergo for apoptosis. To answer your question, yes, we need a multi-pronged approach.
Yes, sir.
Hmm.
And when we are seeing, like I have not many studies,
Hmm.
I have seen some studies which shows that these kind of epigenetics, nutrigenomics,
Hmm.
and what we have seen now, the metabolic modulation
Hmm.
takes the patient so much time. Like கொஞ்சம் மாசம் ஒரு வருஷம் எடுக்கும் அடாப்ட் ஆகிறதுக்கு அப்படின்னு அந்த ஸ்டடி சொல்லுது. பட்
Okay.
மோஸ்ட் ஆஃப் த பேஷண்ட்ஸ் என்ன அவங்க விருப்பப்படுறாங்கன்னா, எனக்கு வந்து சீக்கிரம் சரியாகணும், எனக்கு சீக்கிரம் சரியாகணும் அப்படின்னு போகும்போது they end up with this kind of therapies, immunotherapy, radiation therapy, but they are not aware of like what are the consequences they are going to see. பட் நாம இப்போ இது கொண்டு வரோம், மெட்டபாலிக் மாடுலேஷன் கொண்டு வரோம், இது நம்ம பண்ணலாம், இதனால அவ்வளவு சைடு எஃபெக்ட்ஸ் இல்லை நம்ம சொல்லும் போது பேஷண்ட்ஸ்க்கு வந்து அந்த அவேர்னஸ் இல்லை. தே வாண்ட் லைக் இமிடியட் ட்ரீட்மெண்ட். ஸோ அப்போ லைக் நம்ம என்ன சார் பண்றது?
I don't know how to answer this question, but I think, as I told you, that is a challenge. Okay. See here, one diet plan or one dietary regimen will not work for everybody. The most of the papers which I read so far that says this says this one thing: one size does not fit all. You have to personalize the diet regimen based upon that person's genetic makeup. Some patients will absorb more iron, some patients will absorb less iron, some patients will absorb more folate, some patients will absorb less folate. Okay, already they're having a disease going on in their body. They will not be able to take proper nutrition also. So you have to give this diet in a more feasible manner which they can take it. So there are a lot of challenges here. So it takes time, plus running this multiple omics. Okay, it involves huge cost also. Okay, in India it is an upcoming field. Okay, I think it needs a lot of research on that area, and then we, like I said, it's personalized. Right now this nutrigenomics is not even in the cancer domain, but it is more into the sports nutrition domain. So athletes are undergoing this nutrigenomic, creating their own dietary profile to enhance their performance. Okay, like that, some amount of research should go into this research, into this nutrigenomics. There's a lot of research happening, but a lot of papers has come out. But in India, I couldn't see any paper. I think it is a huge, there's a huge gap there. It's a long way to go, as far as I know.
Yes, sir. I have seen the papers from US, that FDA, and then from UK, and then, as you said, Denmark. Like that in Germany side only, I've seen those papers. As far from India, we have not yet received, or I haven't seen like the papers.
No, I am doing this only for the last 2 weeks. Okay, I want to, to be honest and frank, maybe if I do some advanced search, I will get some papers in India also.
Sure, sir. Thank you, sir. Thank you for answering. Thank you so much.
Thank you. Thank you.
Is there anyone else having some doubts? Then we'll move on to the vote of thanks. Hariharan?
I am here to deliver the word of thanks. On behalf of medicine, I extend my heartfelt thanks to Dr. Jagadish for taking time out of the busy schedule to be here with us today and sharing his expertise with such generosity. I would also like to sincerely thank our wonderful audience for your attentive participation and enthusiasm, which truly enriches every academic session. Finally, I thank the entire organizing committee of medicine for giving me this wonderful opportunity to introduce our esteemed speaker and to be a small part of this wonderful program. Thank you.
Thank you.
Thank you, sir.
Sir, thanks a lot. Thanks a lot, sir.
Thank you. Thank you.
On behalf of medicine, Rahu sir, Jamuna ma'am, we are very thankful for your time.
Thank you for giving me this opportunity, guys. Thank you.
Sir.
Okay. Meet you again in some other webinar.
Sure, sir.
Sure.
Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Bye. Take care.
Thank you for all the participants, and we can leave now.